Welcome back to the deep dive. Today, we are taking on the vascular plant. The blueprint that defines everything from the grass in your lawn to the oldest redwood. We're going to get right into the core of botany to understand how these organisms, which are, stuck in one place, manage to thrive.
It's one of the most brilliant evolutionary compromises out there. Think about it. The basic problem for a plant is it needs things from two completely different worlds at the same time. It needs water and minerals from the soil,
Down below.
and it needs sunlight and CO2 from the air above. Everything we're going to look at today, from organ arrangement to cell walls, it's all a perfect hierarchical adaptation to this dual life.
And that's really the key, isn't it? The structure is all about capturing resources. So our mission today is to map this whole blueprint out. We'll cover the organization, the engine that drives their continuous growth.
The meristems.
The meristems, right. Then how plants grow long primary growth and how they grow thick secondary growth. And finally, the amazing genetic controls behind it all. Okay, let's start with the big picture, the organ systems. It's basically a two-part machine. You have the underground root system.
Right, for anchoring, storage, and absorption.
And then the shoot system up in the air stems and leaves, all about getting up to the light. And they are completely locked in, dependent on each other.
Totally codependent. The roots would starve without the sugars that the leaves make and send down.
And the shoots would just dry up and die without water from the roots. It forces a really efficient design. So let's look at roots first. There are two main strategies, right, depending on the plant group. If you're a eudicote, you often get a taproot system.
That's the one big vertical root. It anchors the plant deep. Think of a carrot or a beet, that's the taproot, and it's also a major storage organ for all those carbs.
And then the other type.
Then you have the fibrous root system, which you see in monocots like grasses. It's this dense, shallow mat of slender roots. It's not great for deep anchoring, but it's fantastic at holding topsoil together.
So good for preventing erosion.
Incredible for it. But no matter the system, the real MVPs of absorption are the root hairs.
And this is where the scale just becomes mind-boggling. Root hairs are these tiny finger-like extensions, and they exist for one reason, to crank up the surface area.
The numbers are just staggering.
I read that a single rye plant, after just 4 months, can have enough root hairs to stretch over 10,000 kilometers if you laid them end to end.
And that's how you maximize absorption when, you can't move to a better spot.
Okay, so moving up to the shoot system.
Yeah.
The stem's main job is just get those leaves up to the light.
Verticality. It's built around nodes, which are the points where leaves attach, and the segments between them, the internodes.
And you see some really wild modifications here, too.
Oh, absolutely. Things like rhizomes, those horizontal stems underground, or stolons runners on the surface. They might look like roots, but they're structurally stems adapted for other jobs.
And the leaves, of course, are the solar panels. So much variation, but the function is always the same. Capture as much light as possible. You see simple leaves, compound leaves.
Or even highly specialized things, like the tendrils of a pea plant that help it climb, or the sharp spines on a cactus for defense. The plant body is just incredibly plastic. It adapts its form to its habitat.
Now, if we zoom in from the organs to the tissue systems, you're saying we can basically group everything into 3 continuous systems.
Exactly. 3 systems that run through the whole plant, root and shoot. Think of it as the skin, the plumbing, and, well, everything in between.
Okay, give us the big 3.
First up, the protective skin is the dermal tissue. In non-woody plants, that's the epidermis, and it's usually coated in a waxy cuticle.
Which is key for preventing water loss, right?
Yeah.
For life on land.
Absolutely essential. If the plant is older and woody, that epidermis gets replaced by a tougher material called periderm.
And the dermal layer isn't just a passive wrapper. It has active parts.
For sure. You've got guard cells that form the stomata, acting like little gates for gas exchange. And then you have trichomes, all sorts of hair-like structures. In some plants, they reflect sunlight, and others, they're like little glands that secrete defensive chemicals.
Okay, system number 2, the internal plumbing. That's the vascular tissue system.
The transport superhighway. It's got 2 parts. You have xylem, which is a one-way street, moving water and minerals up from the roots.
And phloem.
And phloem is the two-way street. It moves the sugars made during photosynthesis, the photosynthates, to wherever they're needed, up to a growing tip, down to the roots for storage.
So the arrangement is different in different organs.
Right. In roots, it's often a solid central core called the stele. In stems and leaves, it runs in these separate strands we call vascular bundles.
Which leaves the third category.
Okay.
Anything else? The ground tissue system.
Yep. Everything that isn't dermal or vascular, this is the bulk of the plant. It does photosynthesis, storage, support. We call the ground tissue inside the vascular bundles the pith, and the tissue outside is the cortex. It's way more than just filler.
So if we go down one more level to the cells themselves, there's this perfect gradient of commitment, I guess, from flexible cells to totally rigid ones.
That's a great way to put it. You start with the most versatile, the parenchyma cells. These are the real workhorses. Thin, flexible walls. They do most of the metabolism, photosynthesis, storage. And crucially, they can still divide and differentiate. They can heal wounds.
Then you get the first level of dedicated support, the collenchyma cells.
Right. These give flexible support to young, growing parts of the plant. Their walls are unevenly thickened, but they're alive and can stretch. They let a young stem bend without snapping.
But for real, hardcore rigidity, that's where sclerenchyma cells come in.
That's the hard stuff. These cells have thick, rigid secondary walls that are full of lignin, which is this incredibly strong polymer.
And to get that strong, they have to make a sacrifice.
They do. They're often dead when they're functionally mature. They leave behind their lignified walls as a sort of permanent skeleton.
And these are things like fibers for rope or the sclerids that give pears that gritty texture.
Exactly.
The transport cells are just as specialized. Xylem has to move water under pressure.
Which is why that dead lignified support is so important. Xylem uses tracheids and vessel elements. They're basically hollow, dead tubes reinforced with lignin. Vessel elements are super efficient, forming these long, continuous pipes called vessels.
And phloem is different. The cells for moving sugar, the sieve-tube elements, they're weird. They're alive, but only just.
They're highly modified. To make room for sugars to flow, they get rid of their own nucleus, their ribosomes, their large vacuole. They're completely dependent on an adjacent companion cell that handles all the metabolic work for both of them. It's this beautiful division of labor.
Okay, so we have the anatomy down. Now the big idea for plants, they just keep growing. This concept of indeterminate growth is pretty wild. How does that blueprint keep running forever?
It's all because of meristems. These are specific regions of perpetually dividing undifferentiated cells.
And this is the opposite of determinate growth.
Which is when an organ, like a leaf or a flower, grows to a certain size and then just stops.
So meristems are the engine. And there are two main ways this engine works: growing long versus growing wide.
Precisely. For growing in length, plants use apical meristems. They're located right at the tips of roots and shoots. This is what drives primary growth in length. The cells they produce mature into the three primary tissues we already mentioned.
But for big plants, for trees, they need girth. They need to grow wide. That's the second type.
The lateral meristems. These are cylinders of dividing cells that run the length of the older stems and roots. They're responsible for secondary growth, which is growth in thickness. And there are two of them, the vascular cambium and the cork cambium.
Okay, let's stick with primary growth for a minute, the lengthening part. How does a root tip push through hard, abrasive soil without getting destroyed?
It has amazing protection. The root cap is like a tiny little helmet of cells covering the delicate apical meristem. It also secretes a polysaccharide slime that lubricates the soil ahead of it.
And the pushing force doesn't come from the dividing cells.
No, it comes from the zone of elongation just behind it. That's where the newly made cells fill with water and expand rapidly, shoving the tip forward.
And once those root cells differentiate, the tissue layout is all about control.
Yes. The vascular cylinder, the steel, is surrounded by the cortex, and the innermost layer of that cortex, the endodermis, is the gatekeeper. It acts as a selective barrier. It makes sure that water and minerals have to pass through a living cell membrane before they can get into the vascular system to be transported up.
It's so interesting that lateral roots emerge from deep inside the main root.
While branches on a shoot grow from buds on the outside.
Why the difference?
Lateral roots have to originate from the pericycle, which is the outermost layer of the vascular cylinder. They have to, because their plumbing has to be connected to the central plumbing from the very beginning. So they have to burst their way out through the cortex and epidermis.
Now, switching to primary growth in the shoot, we get to something every gardener knows about: apical dominance.
The shoot apical meristem at the very tip of the stem is in charge. It produces the new leaves and stem, and apical dominance is basically a chemical signal sent out by that top bud that tells the nearby axillary buds, the side buds, don't grow yet.
It's a strategy to prioritize growing straight up towards the light.
Exactly. And when you prune that top bud, you remove the signal, the axillary buds are released from dormancy, and the plant starts to branch out.
Okay. Inside the leaf, the ground tissue is specialized. It's called the mesophyll.
And it's usually split into two layers. The upper layer is the palisade mesophyll, tightly packed cells to capture the most light. Below that is the spongy mesophyll. It's full of large air spaces that let CO2 and oxygen circulate. It's like the plant's lungs.
And the ports for that system are the stomata.
Correct. The pores, flanked by guard cells that regulate the whole thing. And it's a constant trade-off.
The balancing act.
A huge one. They need to let CO2 in for photosynthesis, but every second a stoma is open, the plant is losing precious water.
Let's shift to stability now. Secondary growth. This is how a thin little stalk becomes a massive woody trunk.
This is the signature move of gymnosperms and many eudicots. Most of that thickening comes from the vascular cambium. It's a thin cylinder of meristematic cells, and when it divides, it produces secondary xylem, which is wood to the inside, and secondary phloem to the outside.
And the buildup of that secondary xylem is what makes a tree so massive.
That's right, year after year of accumulation.
And the pattern of that accumulation is what gives us growth rings.
The seasons are the key. In the spring, when water is plentiful, the tree makes early wood with big, wide cells for rapid water flow.
And in the late summer.
It makes late wood. The cells are smaller, with thicker walls, more for support. The visible line between the dense late wood of one year and the wide open early wood of the next year is the growth ring.
And that's the basis for dendrochronology, studying past climates from tree rings.
That's the science.
So as the tree gets older, the wood in the very center isn't really doing anything for transport anymore.
Right, that inner wood stops conducting water and becomes heartwood. The tree often pumps it full of protective resins and tannins, making it resistant to rot.
So it becomes the structural core.
It does. And the outer, still functioning layers of xylem are called the sapwood.
And the outer protective layer, the bark, is produced by that second lateral meristem.
That's the cork cambium. As the trunk gets wider, the original epidermis splits apart, so the cork cambium forms and starts producing tough cork cells. These cells get coated in suberin, a waxy, waterproof material, and then they die.
So technically, what is bark?
Technically, bark is all the tissues outside the vascular cambium, so that's the secondary phloem, plus the cork cambium and all the cork it produces.
And if that whole outer layer is waterproof, how do the living cells inside get any air?
Through lenticels. They're small, spongy, raised areas in the periderm that allow for gas exchange with the atmosphere.
Okay, we've got structure and growth. Let's talk development. How the plant actually takes its shape. This idea of developmental plasticity seems really important.
It's maybe the biggest difference from animals. Plants can't run away, so they have to change their form to deal with their environment. The example of Cobomba aquatica is perfect. Underwater, it grows feathery leaves. But when it hits the surface, it starts producing these flat, floating pads. Same genetics, different form based on environmental cues.
And we've learned that plant growth is less about making tons of new cells and more about just enlarging the ones they have.
That's a huge part of it. Cell expansion, mostly by taking up massive amounts of water into the central vacuole, is a very cheap way to grow fast. But it's not random expansion, it's guided. The direction of growth is controlled by the orientation of cellulose microfibrils in the cell wall.
A kind of internal scaffolding.
An internal scaffolding that's laid down by the cytoskeleton. It's all very deliberate.
And when it comes to a cell's fate, what it becomes, the modern view isn't about its lineage, but its address.
Exactly. Position-based mechanisms are everything in plants. A cell's fate depends on the signals it gets from its neighbors. The classic example is in the Arabidopsis root. If an epidermal cell is touching two underlying cortical cells, that positional cue tells it to become a root hair cell.
And if it's only touching one?
If it's only touching one, it gets a different signal that tells it to remain a hairless cell. The pattern is created entirely by location.
It's all chemical signaling. And you see these huge shifts, not just cell by cell, but across the whole organism in what are called phase changes.
Yes, like the change from a juvenile leaf shape to an adult leaf shape. The meristem itself matures over time. But the most important, most tightly controlled phase change is the switch from growing to reproducing, making a flower.
And this is where we see that genetic control in its most elegant form, the ABC hypothesis.
Flower development is a switch from indeterminate growth to determinate growth. The ABC model explains how the four parts of a flower form using just three classes of genes, A, B, and C. A genes alone give you sepals, the outer layer. A plus B gives you petals, B plus C gives you stamens, and C genes alone give you the carpels in the center.
And the clever part is the mutual inhibition.
That's the genius of it. The A and C genes shut each other off. So if A is missing, C takes over its territory and vice versa. It creates this incredibly robust, reliable system for building the reproductive organs in the right order.
And so much of this we know because of model organisms, like the little weed Arabidopsis thaliana.
A powerhouse for developmental biology.
Hmm.
Small, fast-growing sequenced genome. It's let us uncover these fundamental blueprints.
So if we step back, what really stands out is this dynamic balance. The incredible complexity of a plant is built through this continuous dynamic growth from meristems.
And structure is just perfectly fit to function at every single scale, all allowing the plant to adapt to its environment.
And for a final thought, it's that contrast we just talked about. The main body roots, stems, leaves is so plastic, its growth is indeterminate, all to survive in an unpredictable world. But the flower, the thing that guarantees the next generation its growth, is determinate, and it's governed by the strict lockdown logic of that ABC gene network.
Which leaves a question for you to think about. If flower formation is so tightly controlled compared to vegetative growth, what does that tell us about the evolutionary priority plants place on guaranteed successful reproduction versus flexible individual survival?
Something to consider the next time you're looking at the plant life around you. Thank you for joining us for the deep dive. We hope this gives you a much clearer map of how a plant is built and how it grows.